This presentation is the second in a series of training manuals. The first was designed for setup and for assembly. The IFL has a number of unique capabilities. It has a white actinic light source that provides closer results when compared to sunlight than using an intense red light source with only a 20% blue actinic light. The IFL has an RGB leaf absorbance measuring capability that closely estimates leaf absorbance when compared to integrating sphere measurements, the standard in such measurements, providing more reliable electron transport rate values, mesophyll conductance values, and CO2 at the site of carboxylation measurements. The IFL has walkaway automation. Normally, with gas exchange tools, you have to worry about a couple of things. So you have to babysit. You have to worry about erga drift over short periods of time, and you have to worry about leaves drying out and becoming inactive in chambers over time. The IFL automatically zeroes out the erga with every measurement, so you don't have to worry about erga drift. It also has a wetter column, which allows you to program in near ambient humidity conditions. This helps prevent leaves from becoming inactive, so you can push a button and walk away. The IFL also has automated post-processing capability, and it calculates values after you run a protocol for LASK, Koch, Yin, or the Flexus chamber leakage protocol. It provides direct readouts of respiration in the day, gamma star, or the compensation point where there's a lack of mitochondrial respiration, GEM or mesophyll conductance, and CC or CO2 at the site of carboxylation. The battery charge life is between 8 and 16 hours at 20 degrees centigrade with the IFL. If you're using the chlorophyll fluorometer in conjunction with this system, the battery charge life is closer to 8 hours. The IFL LC Pro SD system has a built-in auto zero for the erga, and making measurements and the recalibration happen over a 16 to 24 second period automatically. Protocols can last for hours. And with the IFL, you can push a button and walk away, as we've said before. The system has also an ambient PAR sensor above the light source of the IFL. This allows you to duplicate PAR light levels within the chamber, measuring chamber, and it also allows you to lock in ambient light levels on partly cloudy days. On partly cloudy days, measuring chlorophyll fluorescence becomes problematic because chlorophyll fluorescence requires that the leaf be at steady state photosynthesis. And on partly cloudy days, that commonly doesn't happen. So if you lock in the actinic light level and continue to use that for measurements, it eliminates the problem of partly cloudy days. The automated post-processing we also talked about a little bit earlier, but it is something that's built into the instrument. As I said before, the system has a wetter column, but it allows you to control humidity not only below ambient, but also near ambient and above ambient. This allows one to prevent leaf inactivity when a leaf is in a chamber for a long period of time. ADC Bioscientific was the company that made the first commercially available photosynthesis system based on infrared gas analyzers in 1970. And they were the first company to provide the first commercially available open mode design system in 1984. It was revolutionary at the time, and now all competitors use it. ADC also is the first company to use solid-state H2O sensors to replace ERGAs in measuring humidity. Why? Because it measures to 1 millimole, just like ERGAs do for H2O. However, unlike ERGAs, they don't drift over short periods of time, and they don't eat a lot of significant battery power. The idea is that the measuring system can be more portable, have longer battery life, and be lighter weight. Some time ago, it was found that under near saturated light conditions, that it was impossible to completely close or chemically reduce all PS2 reaction centers, even with the most intense saturation flash. As a result, Bernard Jeanty, the man that invented the light adapted measurement back in 1969, and a number of others, developed a method called FM prime correction that provides a reliable measurement under saturating or near saturating light conditions. The IFL has that capability. However, we aren't the only ones that do provide this. The IFL has up-to-date quenching protocols, including the Kramer and Hendrickson lake model protocols and the Neil Baker puddle model protocol. The IFL has a remote infrared sensor to measure leaf temperature underneath the leaf. This provides more reliable measurements and allows temperature curves that are more reliable because it integrates measurement over a larger leaf area. Leaf area temperature measurements can be spotty or patchy under certain conditions, such as drought stress, cold stress, even under low CO2 levels or biotic stress, you can get patchy results. So using the infrared sensor provides much more reliable results than, say, using a thermocouple. The system also allows measurement with a thermistor for smaller leaves or calculated leaf temperature, should you want to use that. The instrument provides a larger leaf temperature measuring range as well. If you're doing temperature curves, you can go to plus or minus 10 degrees centigrade in your work. Temperature curves are valuable when working with GM or mesophyll conductance because GM has been found to vary with leaf temperature. The actinic white light provides data closer to sunlight conditions in ACI curves, ACC curves, and AQ curves. It does require a certain change in measurement protocol, which we'll go through in a bit. The IFL offers an automated modulated light setup capability. The automated system starts with increasing the gain and then increases the intensity. The intensity of the modulated light is less than 1 micromole, and it's designed to detect pre-photosynthetic chlorophyll fluorescence in PS2, antenna fluorescence. And if set correctly, it does not drive photosynthesis. You can still set it manually should you want to, but the automated version is less problematic and it works extremely well. Photosynthesis is comprised of a light reaction and a dark reaction. Chlorophyll fluorescence measures the light reaction, and gas exchange measures the dark reaction. In C4 plants, the relationship between the two is linear. In C3 plants, however, the relationship is generally curvilinear. Why? Because in C3 plants, rubisco will combine with either carbon dioxide or various forms of oxygen. Some researchers use an air tank mixture of 1 to 2% oxygen to minimize the effects of O2. A tank without any oxygen should not be used. The exact correlation between gas exchange and chlorophyll fluorescence is not possible because chlorophyll fluorescence measures the top layers only, while gas exchange measures the entire leaf. For these reasons, the relationship can be messy and complex, according to Rosequist and Van Kooten. There are a few reasons for using an integrated system with chlorophyll fluorescence and gas exchange. One is that you're measuring the light and dark reaction at the same time over the same leaf area. The second is that such an instrument can be a measuring standard for other kinds of measurements. Gas exchange takes a while to take a measurement. And so if the experiment requires larger plant populations, going to faster types of measurements such as chlorophyll fluorescence or chlorophyll content measurement, the combination can act as a measuring standard for such activities. Now we are going to review chlorophyll fluorescence measurements. The primary reason for combining gas exchange and chlorophyll fluorescence is to allow measurement of ACC curves, or photosynthetic rate at the site of carboxylation curves. This requires measurement of GM. These are more reliable than ACI curves. While there are other methods for measuring GM, chlorophyll fluorescence and gas exchange are by far the most cost-effective way to do it. To measure GM or mesoville conductance and determine CO2 at the site of carboxylation, the affordable way to measure GM and CC requires chlorophyll fluorescence. Variable chlorophyll fluorescence only happens in photosystem 2, not photosystem 1, and only with chlorophyll A. Chlorophyll fluorescence from photosystem 1 does exist at a very low level, but it is not variable. In C3 plants, the correlation with gas exchange measurements is curvilinear due to photorespiration. Why? Because in C3 plants, rubisco will combine not only with carbon dioxide, but also various forms of oxygen. As a result, this is called oxidative stress, and it makes chlorophyll fluorescence under oxidative stress delay measurement of plant stress until the stress is more severe. For instance, under drought conditions, it's possible for the chlorophyll fluorometer to take 7 days before it can detect drought stress, whereas gas exchange will detect drought stress after about a day without watering. Under heat stress, again an oxidative type stress, the chlorophyll fluorometer will detect heat stress at about 35 degrees centigrade using the light adapted version, or it will be up to 45 degrees if using FEFM, the dark adapted version. Gas exchange will detect heat stress at about 30 degrees or above. This picture shows where gas exchange occurs and chlorophyll fluorescence occurs. It also shows the locations of CI and CC. In the upper left-hand corner, you've got a C3 plant, and you can see the stoma where air enters the leaf and where the CO2 becomes available to the plant. The intracellular airspace, shown as CI, is one of the values in ACI curves. A is photosynthetic rate compared to CO2 levels at the intracellular airspace areas. ACC curves, on the other hand, are photosynthetic rate compared to CO2 at the site of carboxylation inside the chloroplast. So the CO2 has to diffuse through the cell wall, the cell membrane, the cytoplasm, diffuse through the chloroplast and into the chloroplast stroma where carboxylation occurs. Inside the chloroplast on the thylakoid membrane is where chlorophyll fluorescence occurs. Now, GM is the rate of conductance from the intracellular air spaces to the site of carboxylation inside the chloroplast. And it is affected by species, the resistance of the cell wall, resistance of the cell membrane, the cytoplasm, chloroplast diffusion. It's affected by temperature and CO2 level. Variable chlorophyll fluorescence only happens in the thylakoid membrane in photosystem 2 with QB- reducing reaction centers, shown in the red square. When dark adapted, you end up with minimum fluorescence or pre-photosynthetic fluorescence. Photosystem 2 is completely oxidized because chlorophyll fluorescence and photosystem 2 is an oxidation reduction reaction. And as you add light, QA in the thylakoid membrane is reduced. FM, or maximum fluorescence, occurs when QA-, QB2-, and PQH2 are at their maximum. It's also true that at FM, photosystem 2 reaction centers are fully reduced. This slide shows other reaction centers in the thylakoid membrane. We talked about photosystem 2, and we're going to talk about photosystem 1. Photosystem 1 does provide a certain amount of minimum fluorescence, but it is not variable. So it contributes to FO, or minimum fluorescence. On the right-hand side, you'll see a non-QB- reducing reaction center, PS2. And it is the result of photo damage. It also contributes to FO, or minimum fluorescence. And photo damage contributes to the fact that FEFM and Y2 are reduced because of photo damage. Both the chlorophyll fluorescence and gas exchange measure over the same area. However, under certain circumstances, there's such a thing as chlorophyll fluorescence heterogeneity. It happens under drought stress, cold stress. It can happen under reduced CO2 levels in ACI and ACC curves. And it can happen under biotic stress. What happens is, if you measure chlorophyll fluorescence on different parts of the leaf, you get different results. The advantage of using the integrated gas exchange chlorophyll fluorometer is you're measuring chlorophyll fluorescence over a large area. And so heterogeneity is not as much of an issue as it would be if you're using a standard chlorophyll fluorometer. This slide talks about issues related to C3 plant measurement. Gas exchange will detect plant stress of all different types reliably and early. For instance, it will detect drought stress one day after the stopping of watering. It will detect heat stress at about 30 degrees centigrade, and it will do a good job of measuring all different kinds of stress. As far as chlorophyll fluorescence is concerned, the light-adapted test, Y2, will detect drought stress after about 7 days of stopping watering. Why? Because of oxidative stress, as we discussed earlier. Heat stress, the light-adapted test will detect heat stress at about 35 degrees centigrade or higher. It does a good job on all other kinds of plant stress except for nitrogen and sulfur. And with nitrogen and sulfur, it will not detect stress until the plant is nearly dead. FEFM, the dark adapted chlorophyll fluorescence test, normally detects drought stress after about 7 days as well. However, there's a recent paper that came out that said if the ambient temperature is above 26 degrees centigrade, that FEFM will detect drought stress in C3 plants 1 day after stopping watering. FEFM will not detect heat stress until about 45 degrees centigrade in C3 plants, again, because of oxidative stress. And again, all other kinds of stress, FEFM will do a good job with, except for nitrogen and sulfur stress. And again, it will only detect nitrogen and sulfur stress when the plant is near death. This slide talks about measuring issues for C4 plants. As in C3 plants, gas exchange does a good job with all types of measurements. And since there is no oxidative stress in C4 plants, because there's a CO2 concentrating mechanism, the Y2 and FEFM chlorophyll fluorescence measurements also do a good job with drought stress and heat stress in C4 plants. Essentially, chlorophyll fluorescence does a good job on all kinds of plant stress in C4 plants, except for nitrogen and sulfur stress again. Here, it will only detect nitrogen and sulfur stress when the leaf or the plant are nearly dead. And under those circumstances, if you need a fast way to measure, using a chlorophyll content meter is the best choice. Chlorophyll fluorescence measures the light reaction, but there is a dark adapted test that is extremely popular. It's FEFM. FO, part of FEFM, is minimum fluorescence, a dark adapted value that represents pre-photosynthetic chlorophyll fluorescence, is measured by a correctly and low-level modulated light. FM, or maximum fluorescence, after dark adaptation and after measurement of FO, there's an intense saturating light source, flash, that completely saturates photosystem 2, and it represents the highest variable chlorophyll fluorescence possible. In the IFL, it represents the highest 8-point rolling average over a 0.125 millisecond period. FEFM is a normalized ratio. Basically, the values range from 0.79 to 0.83 on a healthy plant. Lower values indicate plant stress. FEFM is FM minus FO normalized by dividing over FM. This slide shows the FEFM protocol in the IFL. As we said before, a healthy plant would have a value somewhere between 0.79 and 0.83. Lower values indicate plant stress. If you look on the left-hand side of the screen, where there's a red line, you have FM. That's maximum chlorophyll fluorescence. It represents the fact that all photosystem 2 reaction centers are closed or chemically reduced. FO, the lower value, indicates pre-photosynthetic chlorophyll fluorescence, where all PS2 reaction centers are fully oxidized. If you look in the upper right-hand window in yellow, you will also see something that looks like FV/O. That means that instead of normalizing FV over FM, you're normalizing FV over FO. FEFM correlates to gas exchange measurements. FEFO does not correlate to gas exchange measurements. However, it is considered a more sensitive plant stress detector. There's also a light-adapted chlorophyll fluorescence test. We're going to call it Y2, but it goes by a number of names. It can be called delta F over FM prime. It could be called phi PS2, and there are other names. But here we're going to discuss it as Y2. The light-adapted value uses the same formula as FVFM, maximum fluorescence minus minimum fluorescence divided by maximum fluorescence. However, it's under light-adapted conditions. The test was first developed by Bernard Jeanty in 1969. The difference between FVFM and Y2 is that it varies with light level. So if you're going to compare samples or Y2 measurements, they have to be at a similar light level. Part of the Y2 measurement is a calculation or estimation of electron transport rate. It is also called J in gas exchange terminology. Electron transport rate is equal to the Y2 fluorescence measurement times 0.84, or the average leaf absorbance value, times 0.5, or the average ratio of PS2 reaction centers to PS1 reaction centers, times PAR, photosynthetically active radiation or light level. PAR, photosynthetically active radiation, represents the actinic light level on the leaf with values between 400 and 700 nanometers, the visible light range. Our PAR sensors are cosine corrected. This slide shows the IFL using the Y2 protocol. And you can see that the difference between FM prime and FS is much different than it is concerning FVFM in the lower left-hand corner. And that's because the light level raises FS, fluorescence at steady state photosynthesis. FM prime is maximum fluorescence using a saturation flash, just like used in FVFM. FS is again at steady state photosynthesis. Steady state photosynthesis takes between 20 to 30 minutes at high light levels or at low light levels to reach steady state. For a long time, it's been known that if a leaf is under near saturating light conditions, that it is impossible to completely close all PS2 reaction centers or chemically reduce all PS2 reaction centers, even with the most intense saturation flash. To get around this issue or solve this problem, a number of researchers got together, including Bernard Jeanty, the man who invented the Y2 test back in '69, and they developed FM prime correction to solve the problem. And while it's not using an infinitely intense saturation flash, it determines what FM prime would be if an infinitely intense saturation flash were used. This is a diagram of the test, FM prime correction. And while we're not the only company that offers this solution, it's definitely worth using under high actinic light conditions. What they do is they start off with steady state photosynthesis, and they use a saturation flash for 1/3 of a second at 7,000 micromoles. Then they step down to different levels until they reach 5,600 micromoles for 1/3 of a second. At the end, they increase the saturation flash back up to 7,000 micromoles to see if there's any saturation flash NPQ. What they do then is they do a least squares linear regression analysis of the data. The data is the dark red line in the right image. And using the least squares linear regression, they determine what the fluorescence value would be on the y-axis if they were using an infinitely intense saturation flash. And they found that the data correlated extremely well with gas exchange. You have a choice between using the FM prime correction with the IFL or using a square top saturation flash. This is a diagram that compares the screens from the IFL using a square top saturation flash and FM prime correction saturation flash. FM prime is shown with the red line on the left. FS is shown also on the left. On the right, you can see how the saturation flash is divided into three sections. One at 7,000, one with reducing steps down to 5,600, and then back up to 7,000. And you then get the result of Y2. Here it's called PS2 on the screen. The IFL also has the capability to measure or estimate leaf absorbance using RGB sensors under the leaf to measure leaf transmittance and RGB sensors above the leaf to measure leaf reflectance. This system provides a measurement that closely relates to integrated sphere measurements, the standard used for measuring leaf absorbance. The other option, of course, is to use 0.83 or 0.84 as an average value, but this gives you a little more capability to work on reliable measurements. Neil Baker in 2008 stated that if you're going to compare J or ETR values from different leaves, you first must have measurement of leaf absorbance and the ratio of PS2 to PS1 reaction centers. If not, you shouldn't compare ETR values from different samples. An average value is used for leaf absorbance normally, unless you use an integrating sphere. The value that's used on average is 0.84. However, on healthy plants, the absorbance varies between 0.7 and 0.9 in reality. It varies by species, it varies by leaf age, varies by chlorophyll content, and it varies by plant stress and light level. When it comes to the ratios of PS2 to PS1 reaction centers, that also varies by plant type, whether the plant was grown in the sun or the shade, and it can vary by carbon deficiency. 0.5 is used as an average. However, if you want to do some research on the PS2 reaction center ratios for different plants, both the Laisk-Loretto paper in 96 and the Edwards-Baker paper in 93 have a list of values for different species. This slide shows the IFL measurement chamber when it's open, and it highlights an infrared sensor for leaf temperature measurement. The sensor is remote, it doesn't contact the leaf, and it measures about 70% of the leaf chamber, providing a more reliable measurement, even under patchy or heterogeneous temperature conditions. The RGB sensor is also under the leaf and shown here in the picture, and it measures about 80% of the leaf chamber area and is used to estimate leaf absorbance. The white actinic light source of the IFL ensures chloroplast migration, or QM. It also ensures that the xanthophyll cycle works in a more reliable fashion. The high light level avoidance mechanism, chloroplast migration, takes between 20 and 30 minutes to occur. and it only happens under intense white light or intense blue light. The picture on the right shows what chloroplast migration might look like. You've got chloroplasts around the top of the cell or visible from the top of the cell, and they migrate to the sides of the cell, increasing leaf transmittance of light, a photoprotective mechanism. And if you look at measurements made when the leaf is first put under clamp in the IFL, you see that transmittance of the leaf is 2% in blue, 7% in green, and 3% in red. After 30 minutes at high or near saturating light levels, chloroplast migration occurs and the transmission through the leaf increases. Now it's 3% blue, 9% green, and 5% red. So you can see the advantage of being able to measure leaf absorbance. This slide compares measurements at 4 minutes with a white light compared to 36 minutes using white light. You'll notice that not only does the Y2 value measurement change at the bottom from 0.078 to 0.128, but also leaf absorbance changes due to chloroplast migration from 0.83 to 0.76. And of course, this provides a difference in the measurement because electron transport rate, or J, using average values at 4 minutes is 8.2, whereas electron transport rate, or J, after 36 minutes in chloroplast migration is 12.6. The IFL also has up-to-date quenching protocols. You can use the older puddle model protocol by Neil Baker, which has NPQ, QN, and QP, or you can use the newer lake model options that include Kramer Lake model protocol with Y2, YNPQ, YNO, and QL, or the Hendrickson Lake model, which has Y2, YNPQ, YNO, and also features NPQ from the earlier puddle model. When it comes to quenching, the xanthophyll cycle is part of quenching measurements. It's a photoprotective mechanism that protects the plant from blue wavelengths and shorter wavelengths that are more energetic and more likely to damage the plant. Using white light provides a spectrum that is closer to the solar spectrum when measuring quenching values. QE is a measure of the xanthophyll cycle. It measures the fast reacting capability, and it can vary from just a few seconds to minutes in the lab. But if you're measuring field plants, it takes from 4 to 7 minutes in sun-grown leaves to completely adjust. Light that is absorbed by the leaf is used in one of three ways. It can be converted to chemical energy through photosynthesis. It can be re-emitted as chlorophyll fluorescence. And it can also be re-emitted as heat, either regulated heat or unregulated heat. These three mechanisms compete with each other, and when one is maximized, the others are minimized. This slide shows the definitions of the Kramer protocol quenching parameters. The values Y2, YNPQ, and YNO all add up to 1. QL is a measure of open PS2 reaction centers. Y2 is a measure of the portion of light used in photosystem 2 in chemistry. It is also used in the formula for electron transport rate. YNPQ is regulated heat dissipation by photoprotective mechanisms. Y and O is unregulated heat dissipation and chlorophyll fluorescence. NPQ is Y NPQ divided by Y NO. NPQ can be further divided into other plant mechanisms. QE, which is the xanthophyll cycle, or a measure of the xanthophyll cycle. QM, which is a measure of chloroplast migration and can account for up to 30% of NPQ at high light levels. QI is photoinhibition or photodamage. And QT is a measure of state transitions that occur at low light levels. QE, as we've discussed, is a photoprotective mechanism, the xanthophyll cycle, but it only responds, as it does in nature, to intense blue light or white light and not red light. While it can take just a few seconds on indoor plants and algae, it can take up to 4 to 7 minutes in field plants. 80% red light and 20% blue light does not allow the photoprotective xanthophyll cycle to adjust as it does in nature. And since ACI curves are normally done at high saturation levels, except the LACE protocol, 80% red and 20% blue do not allow the xanthophyll cycle to adjust as it does in nature. So you have to question whether or not it provides reliable characterization. This slide shows the pigments of the xanthophyll cycle in the blue box. Essentially, they show the absorption spectrums of zeaxanthin, violazanthin, and zeaxanthin with PSBS. The graphs are in either gray or in black for these pigments. This slide shows the difference between white light and an 80% red and 20% blue actinic light, and how it affects the xanthophyll cycle. You note that after 4 minutes, the electron transport rate is 0.84 with the white light, and it's 16.2 with the high red light and 20% blue. Also, the Y2 values are different. Chloroplast migration, another photoprotective mechanism of the plant, is game-changing. This slide shows a quenching measurement of chloroplast migration. It starts with measuring FO and FO prime and FM, and then the light turns on and it measures under light conditions the changes that occur over a period of time. The first 4 minutes, or first 7 minutes if you're outdoors, is due to the xanthophyll cycle. And you can see how the fluorescent signal changes over that time. But it continues to change for up to 30 minutes. And that change is due to chloroplast migration. Chloroplast moving to the sides of the leaf. And again, from a photoprotective point of view, it protects the leaf from damage. QM, which is chloroplast migration, can account for up to 32% of total NPQ, or quenching, non-photochemical quenching. It's normally measured by quenching relaxation, and that's what the graph below looks like. It's turned on for 30 minutes, and then the actinic light is turned off for 30 minutes, and then there are saturation pulses after the light's been turned off, and you can see how the various mechanisms relax. After 7 minutes, the first saturation flash measures the xanthophyll cycle. And after 30 minutes, it measures chloroplast migration. So you can tell using quenching how successful the various mechanisms are in a particular plant. Next, we will review basic gas exchange measurements. The more advanced gas exchange measurements we'll do in a separate segment under advanced measurements. For a long time, ACI curves and AQ curves were used to characterize plants. More recently, ACC curves have become the standard for characterizing plants. To measure an ACI curve or an ACC curve, it's normally done at saturating light levels. It's recommended that for sun plants, 1,500 micromoles are used, and for shade plants, a lesser value, many say 500 or 800 micromoles of light are used for ACI curves and ACC curves. AQ curves are done at different light levels. So ACI stands for photosynthetic rate compared to intracellular CO2 levels. ACC curves are photosynthetic rate compared to CO2 at the site of carboxylation. AQ curves are photosynthetic rate compared to light levels or PAR levels. This slide shows the differences between gas exchange measurements and fluorescence measurements on the IFL. It takes between 16 to 20 seconds to make a gas exchange measurement on the IFL. However, you need to put the leaf into the measuring chamber, and it has to climatize to the measuring chamber for 45 seconds to about 2 minutes before measurement. Gas exchange will measure all types of plant stress reliably. The light-adapted test for chlorophyll fluorescence only takes 2 to 4 seconds to make a measurement. However, according to experts, chlorophyll fluorescence measurements require steady-state photosynthesis for reliable Y2 measurements. The light-adapted test will correlate in a curvilinear fashion with gas exchange due to oxidative plant stress types, and will correlate in a linear fashion to gas exchange in C4 plants. The light-adapted test will only measure drought stress after 7 days, and it will measure heat stress above 35 degrees centigrade. It will also not measure sulfur or nitrogen stress until the plant is nearly dead. As far as dark adaptation is concerned, dark adaptation is commonly done for 30 minutes, but some people believe in overnight dark adaptation. It again takes 2 to 4 seconds to make an FEFM measurement, and it will detect drought stress in C3 plants when the drought stress is more severe than in gas exchange. However, there is a paper that states that at 26 degrees centigrade or above, FEFM will detect drought stress 1 day after watering. It will only detect heat stress at 45 degrees centigrade and above. And it should not be used for nitrogen or sulfur stress because the leaf needs to be almost dead before it will detect a difference. And like the light-adapted test, it has a curvilinear correlation to gas exchange in C3 plants and a linear correlation in C4 plants. This slide makes recommendations for CO2 levels and light levels for ACI curves for both C3 plants and C4 plants. The recommendations were taken from Evans and Santiago, 2014. You notice that the steps for CO2 are different for C3 plants than they are for C4 plants. Notice also that the light levels are the same. For sun-grown leaves, they recommend 1,500 micromoles, and for shade-grown leaves, 500 micromoles. For indoor-grown plants, it will be substantially less. The steps they recommend are 3 to 7 minutes for each CO2 level, because it takes that long for the CO2 to adjust. With the IFL, we make or recommend a change to those settings, in that the first step should be 30 minutes long. And the reason for that is that it takes up to 30 minutes for the xanthophyll cycle and the chloroplast migration protective mechanism to adjust to the light level of the IFL, and as it does under normal sunlight conditions. Otherwise, the steps are the same for both CO2 and for light levels. For AQ curves or light curves under an 80% red light and 20% blue light combination, Evans recommends dwell times as 3 to 7 minutes using 400 micromoles or ambient CO2 levels at standard ambient temperatures. However, there are changes when you go to the IFL. With the IFL, it takes up to 30 minutes for the xanthophyll cycle and for the chloroplast migration mechanism to adjust. And for that reason, we recommend that the first 2 or 3 steps are at 30 minutes. After that, you can go down to 7 minutes for each step. You're going to have different light curves or different values for sun-grown leaves than you are for shade-grown leaves than you are for indoor plants. Indoor plants will be considerably less. The next couple of slides cover definitions for gas exchange. CO2 reference is CO2 level of air entering the measuring chamber measured by the infrared gas analyzer. CO2 analysis is CO2 level in the air leaving the measuring chamber as measured by the infrared gas analyzer. Delta CO2 is the difference between CO2 analysis and CO2 reference. It's the CO2 level absorbed by the leaf. A is photosynthetic rate of CO2 exchange in the leaf chamber in micromoles or in parts per million. You choose. A is mass flow of air per meter squared of leaf area times CO2 analysis minus CO2 reference in micromoles or in parts per million. CI is substomatal CO2 level in the air openings inside the leaf after air enters the stoma. CC is CO2 at the site of carboxylation inside the chloroplast stroma. CC is equal to CI minus A divided by GM. More definitions. GS is stomatal conductance of H2O in millimoles. Stomatal conductance is reduced or eliminated under drought stress. E is transpiration in millimoles. This changes under drought stress. Humidity increases in the air leaving the measuring chamber. Q is par light level on the leaf surface in micromoles. T is leaf temperature, the temperature on the bottom of the leaf to plus or minus 0.1 degrees centigrade. GM is mesophyll conductance rate of CO2 flowing from the intracellular air spaces through the leaf to the stroma inside cell chloroplasts. This slide shows how mesophyll conductance works and its limitations. After air enters the stoma, the intracellular air and the intracellular CO2 diffuse through the cell wall, through the cell membrane, through the cytoplasm, through the chloroplast, and into the chloroplast stroma, where CC occurs, or CO2 at the site of carboxylation occurs. The rate of conductance from the intracellular air spaces to the chloroplast stroma is called GM, or mesophyll conductance. Stroma is the area surrounding the thylakoids. Description of gas exchange, measurement of CO2. The infrared gas analyzer measures the absorption of infrared in a tube due to the presence of carbon dioxide. The reading is then linearized and scaled according to the calibration set after manufacture and during span calibrations every year or two. Next, there is compensation for changes in atmospheric pressure. The IFL measures atmospheric pressure. Also, the presence of water vapor in the infrared gas analyzer that's measuring CO2 causes pressure broadening. Water vapor readings are used to compensate for this measurement. Finally, leaf transpiration causes the net volume of the air leaving the leaf chamber to be higher than when entering. This volume increase tends to dilute CO2 concentration, causing analysis CO2 measurement and analysis of H2O measurements to be lower. Dilution compensation removes this effect so that delta CO2 and delta H2O values reflect the differences due to absorption by the leaf and not transpiration. This compensation is only applied to the analysis rating. This is a diagram of how an infrared gas analyzer works. It has an infrared light source on one end, a measuring tube with an air inlet and an air outlet, and then it has a filter and an infrared detector. The ERGA on the IFL goes through a measuring cycle. First, it measures air entering the leaf chamber. Then it measures air that's leaving the leaf chamber. And then it measures air that has been scrubbed of all CO2 to zero out the ERGA. It repeats this process, and it takes about 20 seconds, or 16 to 20 seconds to complete the cycle. This slide talks about measuring H2O, or humidity. Normally, in the past, ERGAs were used to measure H2O, but more recently, solid-state sensors have been developed that equal the quality of infrared gas analyzers for H2O measurements, not CO2 measurements, but H2O measurements. As a result, ADC Bioscientific has replaced the infrared gas analyzers for H2O measurements with two high-quality Honeywell solid-state humidity sensors. They measure to the same accuracy as ERGAs. They'll measure to plus or minus 1 millimole, just like infrared gas analyzers do. Because of this, it no longer makes sense to use ERGAs because ERGAs drift over short periods of time. They require zeroing. They eat a lot of battery power. They're a lot heavier. And as a result, EDC has replaced the ERGAs with the solid-state sensors. The readings are linearized and scaled during span calibration, and they drift very, very little. These are measurements required for a reliable photosynthesis measurement. Barometric pressure must be measured by the instrument. Leaf area. The leaf area of the chamber, the broadleaf chamber, is 6.25 centimeters squared. If the leaf covers the measuring aperture, then no measurement is necessary. However, if the leaf is smaller than the measuring aperture, then one has to measure the leaf area and enter the value. Flow rate of the air over the leaf surface is measured. It can be adjusted. It's normally set at 200, but it can be adjusted up or down. Leaf surface temperature. This is measured by an IR sensor below the leaf. PAR light level near the leaf surface is measured by PAR sensors. CO2 level of air entering the leaf chamber is measured with the infrared gas analyzer. CO2 level of air leaving the leaf chamber is measured with the infrared gas analyzer. Water vapor pressure entering the leaf chamber is measured by a humidity sensor. Water vapor pressure leaving the leaf chamber is measured by the humidity sensor. Erga drift over short periods of time are automatically eliminated because the erga is automatically zeroed with every measurement using soda lime. This is a checklist for making reliable basic gas exchange and chlorophyll fluorescence measurements. You have to make sure that all the chemicals used in the console are adequate or less than half used. Allow 45 seconds to 2 minutes for the leaf to climatize to the measuring chamber. If ambient light levels will be used, make sure that the leaf angle relative to the sun stays the same. Set the illumination to track ambient or lock the current measuring value into the IFL light source to eliminate the effects of partly cloudy days. Make sure that the leaf covers the foam gasket of the chamber. If not, the leaf area must be measured and input into the IFL. Make sure that the air going into the instrument has a stable CO2 level over short periods of time. Use a plastic tube that dangles near one's feet, or use an air probe that comes with a system, or use a backpack with a box in the backpack with the plastic hose connected on one end with tape and a hole on the other end. This allows time for air mixing to eliminate fast changes in CO2 levels, which can cause errors. If photosynthesis levels are low, try increasing the flow rate or using the wetter chamber. The chlorophyll fluorescence values and gas exchange values are measured when the measurement button is pushed or as scheduled by inputting a routine. Chlorophyll fluorescence values Y2 and J require steady state measuring conditions, usually 30 minutes at the same PAR light levels. It is not necessary to manually zero the erga or match ergas. The IFL uses a single erga that automatically zeroes out drift with every measurement to extend battery life. This is a checklist for reliable ACI curves and reliable AQ curves with the IFL. When measuring ACI curves, use 1500 micromoles of light for sun-grown leaves and 500 micromoles of light for shade-grown leaves. Much lower values should be used for indoor plants. The first step should be illuminated for 30 minutes to allow for steady state lighting conditions. The IFL uses a white light diode for actinic light. It takes the Xantheville cycle up to 7 minutes to adjust, and the chloroplast migration takes from 20 to 30 minutes to adjust. When measuring AQ curves or light curves, the first 2 or 3 steps should last for 30 minutes to allow for chloroplast migration. As steps get lower, chloroplast migration has relaxed. After that, 7-minute steps are recommended for sun-grown leaves. Shorter times can be used for shade-grown leaves. This ends the basic training on the IFL. We will now go to the advanced training, which allows training for measuring ACC curves, respiration in a day, and gamma star measurements, along with some other important things.